So today I'm going to tell you a little bit about these quantitative and multiplexed in vivo functional cancer genomics platforms that we've developed at Stanford. So just a quick disclosure, all the work that I'm going to tell you about today was done during my time as a grad student at Stanford in the genetics department with Monty Winslow in collaboration with Dmitry Petrov's lab, but just in the last few months we have started a company based on this platform. So over the past decade, large-scale cancer genomics efforts have really almost comprehensively cataloged the somatic alterations that we see in human cancer patients. So this is a somewhat old figure of lung adenocarcinoma cancer genome sequencing. And what you can see here is the top most frequently mutated genes on the y-axis and different patient samples on the x-axis. And you'll notice that it's extremely heterogeneous, and as you start going down this list, you very rapidly get to very low frequency alterations. The question here is, we have all of this cancer genomics data. What are these variants actually doing? What's the actual functional goal of these? So that's really what we focus on in Monty's lab at Stanford. So there's a number of systems that sort of fit into broad categories to address this, these sort of functional genomics approaches. You can, of course, use cell lines in a dish. Those are particularly useful when you're trying to work at scale. They're very easy to scale, but they don't have an in vivo microenvironment. There's no immune system. It's typically very difficult to control the genomic alterations, at least the background alterations you're dealing with. They typically are quantitative, which is an advantage depending on how you use them. There's also transplantation models. So these are great models if you're particularly interested in using human cells, and the sort of humanness of the cells is what you care about. But again, we can argue about whether the in vivo microenvironment is correct when you're transplanting these cells, for example, sub-Q on the flank of a mouse. They're typically not immunocompetent. Some people are working on trying to develop immunocompetent models. It's very difficult to control the genomic alterations. You sort of get what you get from the sample. And they're definitely not scalable or quantitative. So the next sort of set of mouse models are these genetically engineered mouse models, where you go into a mouse and genetically engineer an endogenous locus to create typically a conditional mutation that can be activated later in the adult somatic cells of that mouse. So in that context, you're really going in and creating a tumor in the right microenvironment. These models are fully immunocompetent, and you genetically engineer these mice so that you're controlling a very specific set of alterations. However, they're totally not scalable and quantitative. So the goal for sort of my work at Stanford was to sort of get the best pieces of these models into one system. So how can we have an accurate system that's also scalable and quantitative? So diving in a little bit further into these genetically engineered mouse models, the way that these typically have worked is that you engineer into these mice some sort of conditional allele, like, for example, here I've shown this KRAS lock-stop LOX G12D allele that's very common in sort of the field of cancer biology, where in the endogenous KRAS locus, we've engineered in this lock-stop LOX cassette that makes this allele inactive until you get expression of a Cre recombinase that recombines out that stop cassette. And typically the way that this is done is you deliver a virus expressing Cre to a particular tissue type, so for example to the lungs of this mouse, and cells that are transduced by that virus will integrate that viral genome, express Cre recombinase, and then lead to activation of, in this case, KRAS G12D. So at the end, those cells that are transduced may or may not form an actual tumor in a sort of a de novo fashion within that mouse. So the problem is that if you want to study new alterations here, you need to go and create many different mice. And the other issue is that the way that these mouse models are typically analyzed at the end of these experiments is not super quantitative. Like in our lab, a grad student will spend hours, or undergrad will spend hours looking at sections and circling things on ImageJ, or weighing the lungs in mice that have this alteration versus mice that don't. And that's the readout. That's how you tell what the function of that gene is. It's not very quantitative. It takes a lot of time. So these systems are quite slow and expensive to generate. It's not scalable. If you want to study new alterations, it's difficult. The analysis methods are not very quantitative, and there's a lot of variability from mouse to mouse. So it's really not a tractable system to translate all of that human cancer genomics data that I showed at the beginning into actual function, so ascribing function to these different alterations. So the first thing we did to try and tackle this was collaborate with Dmitry Petrov's lab at Stanford, who actually does totally different research in evolutionary genomics. So they use yeast as a model organism. But what they had figured out is that if they barcode the yeast using systems similar to what Paul described, they can study the clonal evolution of many different strains of yeast in parallel using next generation sequencing. So actually we had like a very sort of simple idea to add a random barcode to these lentivectors. So now every lentivector, every lentivirus in your pool has a different unique barcode, and every cell that's transduced by lentivirus gets a stable random DNA barcode. Sort of tags individual cells that get this lentivirus. So then that lentivirus also expresses CRI, it activates KRAS, and if that cell turns into a tumor, it actually amplifies the barcode since it's integrated into the genome. So if you start with a single cell with a unique barcode and you go to 10 cells, you have 10 copies of that barcode. If you go to 10 million cells, 10 million copies of that barcode. So what that lets you do is actually just take the entire bulk lung, not dissecting any individual tumors, just take the entire bulk lung, extract DNA, do PCR for this barcode region, and do Illumina sequencing, and you count the number of barcodes of a specific sequence and stack them together to get the number of cells that existed in each of those tumors within that mouse. And the way that we can convert from reads to actual cell numbers is by spiking into our Illumina reads a particular set of a cell line that has a unique barcode, and we put in a certain number of cells. So for example, if we spike in 500,000 cells into our lung sample and we get a certain number of reads, we know that that is equivalent to 500,000 cells. And so you can back calculate the absolute number of cells that's in every one of these tumors in this mouse just through a single sequencing reaction. So the next thing was, well, now we have this system where we can quantitatively measure the size of every one of these tumors in these mice, but it's still not scalable. We still need to go and make new genetically engineered alleles to be able to study these different alterations in cancer. So what we did next is to design this knock-in Cas9 allele. So now this mouse has a conditional Cas9 gene in its sort of germline. And if you deliver a virus now that has, in addition to this barcode and CRI recombinase, a pool of different guide RNAs, what you get is tumors that have oncogenic KRAS expressed plus inactivation of one of the genes in your library. So you're creating this sort of a double mutation where you have oncogenic KRAS, loss of P53, for example, or in a different tumor, loss of ATM. And this is all happening in the same mouse. So you can get, in this case, we did an experiment where we can get 11 different flavors of tumors within a single mouse. And at the end, we can sequence based on this random barcode, as well as another barcode that's directly associated with each of those guide RNAs, to figure out the genotype and size of every tumor within that mouse. So just to give you a sense of what the data looks like, this is basically raw data after counting barcodes that we see, and this is actually from a single mouse. So from a single mouse, we can find all the P53 deficient tumors, or at least the tumors that received a guide RNA targeting P53, and the size. So on the y-axis, that's the number of cells in every one of those tumors, where each dot is a tumor, and the size of the dot is proportional to the size of the tumor, across all of these different genotypes. So in these mice, we can actually generate many clonal lesions, as you can see. We can generate about 2,000 different clones within a single mouse. So when you aggregate all of this data together, you actually have a lot of data points per genotype. And it allows you to actually fit mathematical distributions to these tumor size profiles to understand a little bit of the underlying biology for each of these genotypes. So this is an example of a little bit more processed data where we've taken the tumor at a particular percentile within each of those distributions and normalized it to guide RNAs that are in each mouse that are inert, that are functionally should do nothing. What you can see here is that For example, LKB1 all the way over here on this side creates much larger tumors all the way from the 50th percentile through the 95th percentile. Whereas some genotypes actually don't seem to be tumor suppressive at all. So if you inactivate those, you don't get much of a shift across the entire distribution relative to these inert guide RNAs. So this really gives you sort of a quantitative metric of the tumor suppressiveness of every one of these genotypes. In this particular experiment, we found SETD2 and RBM10 were these novel tumor suppressors of lung cancer that hadn't been identified before. So this is another representation of the same data, sort of simplified into 3 categories. So now we can classify these tumor suppressors, or putative tumor suppressors, as strong, moderate, or not significant in terms of their tumor suppressive ability. So you can see here SETD2 and LKB1 are very strong tumor suppressors. There's another category of sort of moderate tumor suppressors, and then some that, at least in this experiment, we found is not significantly driving tumorigenesis. The other thing you can do with this system is that you can just add in more flox alleles that people have developed over the past couple decades. And then what you are able to do is look at triplicate mutations. So in that KRAS G12D mouse, if you also have a P53 flox allele where with expression of CRE you inactivate P53, plus your panel of tumor suppressors, you can build this matrix of the effects of particular tumor suppressors in addition to these other coincident alterations. So this gives you sort of an idea of how these mutations interact with each other. And there's some interesting data, at least that I thought came out from this. So for example, if you look at the mouse that has a KRAS G12D driven tumors, P53 deficiency, and RB1 inactivation, you see that that's a very strong set of, very tumor suppressive set of genes. So when you inactivate both of them, you really get lots of tumor formation. That's that dark blue there, suggesting that that might be selected for in human patients. And if you look at human patients, it is actually supported in the data. So this is some human cancer genomics data, where if you look at the co-occurrence of P53 alterations and RB alterations in people, they actually do preferentially co-occur more than you'd expect by chance. There's also some other examples where it's sort of the opposite, where if you look at LKB1, for example, LKB1 by itself in the context of KRAS driven tumors is a very strong tumor suppressor. And SETD2 is a very strong tumor suppressor as well. But when you combine them, you have LKB1 deficiency plus SETD2 inactivation, there's no added benefit, suggesting perhaps that these 2 genes are in the same pathway or doing something redundant. So there's no added benefit to having these. So what you'd expect in humans is that these would be mutually exclusive, since adding that second alteration adds no benefit, and that's indeed true. We see that it's preferentially mutually exclusive in the human data. And actually, if you take this entire grid and correlate it with human data, it does align pretty well. So I hope I've convinced you that this platform it's in the in vivo microenvironment in this autochthonous setting, and that this is a more scalable and quantitative system to start studying these the function of these genetic alterations. But there's one thing that's sort of missing, which is if you look at this list of genes, everything that I've talked about so far has been some oncogenic driver in germline in the mouse plus inactivation of a tumor suppressor using CRISPR. So we're missing, in terms of our scalability, the ability to test activating point mutations in oncogenes. We can't use this CRISPR system to do that. Like for example, in KRAS, of course, there's this KRAS G12 and G13 hotspot where 97% of mutations in KRAS occur. And using the CRISPR system, it's super hard to model this, at least the system that we had designed. What's more interesting is that if you look at these particular loci, like these hotspots, the mutations that occur at those hotspots are quite diverse. So for example, for KRAS G12 and 13, you see a very diverse spectrum of different mutations that occur there. So G12C is the most common in lung cancer patients. G12D and V and A are also very common. But it's very diverse. So when we're using these mouse models that are KRAS lockstep lox G12D mouse to study lung cancer, This would be totally fine if all of these mutations actually have the same sort of biological outputs in response to therapies, but it's not if that's not the case. And for lung cancer, the whole field is using KRAS G12D, including our lab, even though it's not the most prevalent KRAS variant in lung cancer. And the reason that we do that is just because Tyler Jackson, Dave Tuvison made this allele back in the early 2000s, and so nobody wanted to make another genetically engineered allele because it's too hard. So what we thought about is maybe we can adapt this platform that we've developed to do to model these activating point mutations. And the way that we thought we might be able to do this is to, in addition to delivering guide RNA and CRE, to deliver a template for homology-directed repair. So to do that, we had to switch away from lentivirus that has a single-stranded RNA genome and switch to AAV that has a double single-stranded DNA genome. Since then, we could use that DNA potentially as a template for homology-directed repair. So in collaboration with a postdoc in the lab, what we did is we created this AAV vector that has a KRAS homology arm with particular point mutations at position G12 and G13. And we made this as a pool so that we actually have, we can introduce all KRAS codon G12 and G13 non-synonymous point mutations. The other thing we added to retain our ability to quantify things accurately is silent mutations in the downstream codons that act as a barcode. So because we're actually going into the endogenous KRAS gene, we can't just have a random barcode. It needs to be in the sort of wobble bases of the downstream codons. So this is how we tried to get around this. And then the experiment's very similar to the previous experiments, where we take this virus, we deliver it to mice that have Cas9, this time with no driver oncogene. And if the system works, then a cell that's transduced by that virus will activate Cas9, and the guide RNA targeting KRAS creates a break. And that template for homology-directed repair gets used to actually create these different KRAS variants. So after sequencing, what we see is that actually the number of tumors that we get from each of these different variants, even though we started with a pool where the variants were quite even in representation, show up at very different frequencies. So for example, in this lung cancer model, we see G12D and G12R and G13R actually as sort of potent oncogenes, potent driver oncogenes, whereas G12V and A and C are sort of more moderate, and the other ones we couldn't call as sort of oncogenic. So this data actually lines up pretty well, except for a couple of the Rs, with what you see in human non-smoker lung cancer. It's thought that the prevalence of G12C is due to actually the mutagenic assault from tobacco carcinogens. So you see G12C is by far the most common variant in smokers, but we're sort of modeling non-smoking cancer here. So what I've talked about is sort of these two platforms where we can take these barcoded guide RNA libraries to either study tumor suppressor loss in the context of a particular driver oncogene, or to do this homology-directed repair to be able to study activating point mutations in oncogenes. There's many other types of alterations and methods of perturbing cells that we haven't been able to achieve yet. For example, working on trying to understand how the temporal sequence of alterations impacts tumorigenesis and response to therapy is still a challenge. It's still hard sort of technically to be able to create alterations in sort of a temporally distinct fashion. Also doing transcriptomic and epigenomic alterations as opposed to just DNA-based alterations is hopefully a next step we'll get to. There are many more complex genomic alterations you see in cancer, like chromothripsis, where you get shattering of chromosomes, or copy number variation, especially whole chromosome copy number changes, that are very hard to model currently. Hopefully we can get there soon. And the other thing is just trying to apply the system to study the response to drugs in sort of a more comprehensive way. So with that, I'd really like to thank a lot of people at Stanford that helped with these projects. sources of funding. I'd really like to thank Select again for the opportunity to talk, and I'd like to thank you guys for taking the time to listen. If anybody has questions, I'd be happy to take those.